I can just unmute. You're going to have to get the share. Share. Here. Boom. Okay. Let me know. You hit record and then I'll start. Hi, everybody. Thanks for joining this morning for a special webinar from the Z Evolution series. I'm Jim Holland from the USDA in Raleigh. I hosted Z Evolution this past semester. So if you're new to this Z Evolution series, here's some information. We meet every Wednesday at 11 Eastern time. Speakers present mostly unpublished work on maize evolution and adjacent areas, including plant genomics and genetics. We've hosted about 250 seminars since it started in 2019. You can check some of them out in the YouTube channel here. You can contact Jeff Rossibar or Ruben Ryan Alvarez if you want to join it and you don't have the Slack channel information. So today we're here because of Barbara McClintock, a hero to maize geneticists because of their groundbreaking work on cytogenetics and transposable elements. The Maize Genetics Cooperation, which is the organization comprised of and representing the maize genetics community worldwide, established the McClintock Prize for Plant Genetics and Genomics to recognize both Barbara McClintock and active scientists each year who represent the most creative minds and productive scientists in the study of genome structure, function, and evolution in any plant system. The McClintock Prize in 2025 was awarded to Ed Buckler. Normally, the prize winner attends the Maize Genetics Conference and presents a talk in the year that they're awarded the prize, but Ed was unable to attend the conference in 2025. Thus, we're presenting the special Z Evolution seminar to give a wide audience an opportunity to hear Ed present his research. Ed received his bachelor's degrees in both biology and archaeology from the University of Virginia. He then got his PhD in biological sciences from the University of Missouri, Columbia, advised by Timothy Holtzford. Then he moved to NC State for a postdoctoral stint with Bruce Weir and Michael Peruganen. He was hired by USDA ARS in Raleigh to work on maize genetics in 1998. I joined the same unit a year later, so I was Ed's colleague here at this time. Ed moved to a USDA ARS position in Ithaca, New York, affiliated with Cornell University in 2003. Here's the list of chapter titles from Ed's PhD dissertation titled Maize Domestication and Molecular Evolution. I found this list pretty interesting because you can see Ed set out right at the beginning of his research career some themes that marked his later work and contributions. And that sort of the keywords I pulled out of this that I see coming up over and over again later are rapid and inexpensive methods, evolution, climate and plant ecology, and maize genome structure and function. So now I'm going to attempt to condense Ed's about 30 years career into 3 minutes by grouping them into approximately 5-year phases where I see common themes emerging. From the very beginning of his career, you can see that Ed was developing foundational techniques, data sets, populations, and understandings that he would use to build and expand upon. In this first phase, Ed and colleagues used DNA markers to clarify the relationships among maize lines and between maize and its wild relatives, the teosintase. He also performed the first association analysis in plants and described linkage disequilibrium at a whole genome scale. The next phase expanded association analysis into genome-wide association studies, the GWAS as we call it. For this to become a broadly useful technique, Ed and his team established a core diversity association mapping panel for maize, developed the statistical methods to perform GWAS while correcting for the confounding effects of population structure, and developed user-friendly, freely available software to perform the analyses. And meanwhile, he envisioned and guided the building of the maize nested association mapping population, which facilitated multi-population linkage mapping for the first time at large scale. In the next 5 years, Ed and his group expanded these resources by joining deep resequencing of key population founder lines to make the HAP map, and broad population scale genotyping by sequencing, or GBS as you hear it sometimes. This culminated in the joint linkage association mapping studies, where deep sequence data on the 26 NAM founders was efficiently and reliably projected onto the approximately 5,000 NAM progeny mapping lines, enabling in some cases gene discovery for complex traits through first pass genetic mapping. Having established these powerful tools for genetic studies in maize, Ed and his colleagues moved on to understanding more deeply what features of genomes could be related to population genetics of ZEA and phenotypic consequences of different genome features. Ed's most recent career phase has been marked by expanding genomic comparisons beyond the genus ZEA and using deep learning techniques to identify functional features of plant genomes conserved or altered by evolution, and then bringing this information back to bear on maize improvement with the CIRCLE project that aims to improve the cold tolerance, nitrogen use efficiency, and nitrogen remobilization to reduce fertilizer inputs to corn and make the crop more profitable and sustainable for farmers. I think Ed will be the first to say that this incredible record of research achievements was possible only with the help of collaborators. He has said that he's worked with about 2,000 collaborators worldwide. Indeed, a major part of Ed's success, in my opinion, is his generous and wide sharing of ideas, materials, and credit. He's a great collaborator. This has resulted in almost 100,000 citations of his work to date, and is also reflected in his record of ambitious, well-organized, and productive grant projects. And finally, another great impact of Ed's career has been the cadre of students and postdocs that he has mentored who represent major contributors to plant genetics, genomics, and breeding research worldwide. Ed's won too many major awards to list, but here's a few. Notably, he was elected to the National Academy of Sciences in 2014. And Ed is so amazing, the National Academy had to create a new award, the NAS Prize in Food and Agriculture Sciences, for which he was the first recipient. And of course, he was awarded the McClintock Prize for Plant Genetics and Genome Studies in 2025, which is why we're here today. I'm speaking personally as one of the many collaborators who have been tremendously influenced by and made more productive by working with Ed throughout much of my career. He's a great person as well as a great scientist. I consider myself extremely fortunate to have been his immediate coworker during the early years of his career and to continue collaborating with him today. And with that, I want to turn it over to Ed to tell us about his career in maize genetics.
Well, Jim, thank you so much for the incredibly generous introduction. And as I want to share with everybody, what some of the amazing research we've been able to work together on over the years. So I will, first off, receiving the McClintock Prize is certainly one of the greatest honors ever in my life. And I'm just so thrilled to do this, but it really has been that collaboration and working with so many of my friends and colleagues over the years that's been so important. So I'm going to give you the spoiler right now of why do we do maize genetics? It's important science with an incredibly global impact. And that's why I think we're all involved in that, and that's kind of the why we would do it all together. But I then want to start out with a little bit of a personal perspective on how I got into maize genetics, because we each have our own path into maize genetics, and then working together, we can do so much more. I want to mention that McClintock today inspires me every day. Of course, there's that famous picture of her by the McClintock shed. I was walking by on my way into the field. There's her field, her shed. I look out my window right now. I can see essentially the place that the 1932 International Congress of Genetics was there, and McClintock's in that picture. It's that Congress that actually outlined a lot of the genetics that we as a community are working together to figure out and unify. So how did I get into genetics? Well, it actually started with my family. Like so many of us, we have supportive families that allow us to go explore things. My mom was interested in microbiology, my dad in computers. And obviously a lot of my skills and talents fall between those two. And then I married a wonderful maize geneticist in graduate school who's been an incredible support for all the future science I did. Essentially, I got inspired by biology on the hillside of a mountain, or on the side of a mountain, where I got to actually see ecosystems change from a grassland as a forest took it over. And then we would just throw everything we kind of found underneath an old microscope to understand what was going on there. At the same time, I was in that very first wave of kids that got to play with computers and at home. And I started to realize that, wait, genetics is programming with life. And that really early on inspired me that I really wanted to do genetics. But how did I become a corn person?
I think folks cannot see your screen clearly.
What can they see?
It's like gray. The slides are not clear.
Oh.
But as you project this, Ed, it looks clear now. Like so I don't know what's going on.
Let's try again. It was working before. Is that right, folks? Or Jim?
Beautiful.
All right, now you guys see it, okay?
Yeah, perfect.
Okay. Hey, by the way, there's McClintock, okay? Do you guys see this now?
Yes, much better.
Yep, looks good.
Okay, very good. Okay. All right. I think I was right here. Yeah, yeah. So the question of how did I get into maize genetics? Well, I had an incredible high school history teacher who actually spent a whole semester on archaeology. And archaeology got me interested into the questions of how do we, where do we come from, and how do we feed ourselves? And it was there that essentially got me into my interest in corn. As Jim mentioned, I went to the University of Virginia, got this double major in biology and archaeology, had two great advisors there, and Steve Plog and Mike Timko. Then went to the University of Missouri, and both Tim Holzford and Debbie Pearsall and were teaching me a lot about population genetics and archaeology. But and I think I fell in love with the Midwest and with corn and prairie ecosystems. But I also learned how different genetics is from molecular biology. And of course, we want to unify them, but that those are really different ways of looking at the world. And it was at Missouri that I really started to learn about maize genetics. I had some incredible mentors there. Jim Burtzer, Karen Cohn, and Ed Coe were all really key in teaching me about maize. John Dobley was mentoring me from afar. And then when I got to NC State, it was Charlie Stuber and Major Goodman who really taught me so much more about quantitative genetics and maize diversity, and are really the key mentors there. But at the end of my PhD, I was, really enjoyed what I was learning on evolution. But, I was talking to my barber, and I now have the barbershop test. And I was trying to explain to him why I was spending his taxpayer dollars. And I was having a little bit of trouble doing that. And so as I shifted my career going forward, I really wanted to ask this question. And I always ask myself this: why do you do this science? And can I explain it to my mom, my dad, the barber, or the local farmer? And I think there's an infinite number of ways we can do our science, but we do need to be able to explain why we do our science to the people who are paying the bills. And then I moved to NC State, started my USDA position. And again, I had four really amazing mentors: Bruce Weir, Major Goodman, Michael Prugan, and Trudy McKay, where we got started in some of these different areas and kicked off association mapping. But it's then, how do we do that work? It was really then nearly 200 amazing lab members and leaders across the next 25 years that allowed us to do a lot of work that the prize is being rewarded for. And I've also had this incredible group of network across ARS where we were working together to be able to put together these big projects, both out in the field and informatically, in order to make this happen. And as Jim mentioned, it was a lot of different collaborators internationally within the nation that allowed this work to be done. And meeting and working and seeing the world through these 2,000 different collaborators' eyes was so informative to everything we've done. And of course, none of this could be done without funding support. And these are the major groups who funded me throughout my research. But behind each of those institutions were program managers who had a vision, were trying to make connections between groups, and so that these things could be achieved. So let's get back to maize. Maize is pivotal because its photosynthesis is pivotal. And essentially, it fixes anywhere between 1% and 2% of global carbon every year. It occupies a little bit over 1% of all the globe's land, 4% in the US. And if we ate corn directly, it produces enough calories to support 67% of the world. Direct economic impact is about $500 billion, and its indirect impact is measured in the trillions. And so fundamentally, that's why we work on corn. And so what I'm going to be telling you about is how we've been developing, I think, the right set of tools to use this even better. The story of how maize went from that ancestral prairie ecosystem to modern agriculture, what we've learned in some of these areas, and then some of the ways we can envision a 21st century corn. So let's start out with tools.
You can ask me because I don't know where he stores these, and I think
somebody could silence them. So in terms of building these tools, Barbara McClintock did an amazing job of, in order for her to make her discovery, she had to build various tools, whether it was ways to mark and track chromosomes, or in order to follow the diversity of various types of germplasm. And essentially, building the right set of tools is essentially always our microscope that allows us to see in new and different ways. And of course, she used this to understand various aspects of segregation, meiosis, transposons, and genomic diversity. And actually, it was Barbara McClintock's book on the chromosome constitution of the races of maize that during my PhD got me into meiotic drive and also really got me into understanding germplasm diversity. And so key to that, germplasm really was at the heart of everything I've been able to do since then. And because it really captures the heart of adaptation, breeding, and segregation. And as Jim mentioned, essentially we worked through various germplasm pools, whether those were historic inbreds, the NAM population that gave us controlled segregation. We got to see the impact of what breeders had done, looking at the NPGS, germplasm, and XPVPs. And then we got to see how people all across the Americas adapted maize to various locations through the CIMMYT collection and the Seeds of Discovery. But essentially, each of these germplasm pools gave us a new insight into the way we see the world. But we also need to relate that diversity to the phenotypes that we see around us. And essentially there, the statistical and quantitative machinery to relate genotype to phenotype became really key. And we built some key tools to make that possible. Sequencing, of course, drove this. We certainly live in the golden age of genetics, where we can now look at individual base pairs for any genome we would desire. And we can do that now, of course, for a few $1,000s. But if we want to understand how adaptation works, we need to drive it for breeding. We also need to combine that with approaches that allow us to get down to these cheap, low-cost areas. And that's where things like genotyping by sequencing were really key. So to combine that beautiful understanding, we now have the genome down to that $10 or so cost. And then software is really where people can really use these two different tools, the genomic tools, the statistics. And we've had some great teams working together to put together those statistical tools allowed to do G2P connections. And what's been also incredibly rewarding is that people have found that things like Tassel have been really useful tools for teaching both undergraduates and graduate students about how to look at plant diversity. And so where we're going now, as Jim mentioned, we're trying to go, starting to work across species. And when you start to try to make that G2P map across species, this is where these genomic large language models, which are allowing us to map the various portions of the central dogma, are becoming really important. And here we're starting to combine the knowledge and evolution across 65 different plant species, the functional diversity in a few of the model species to model out the central dogma so that we can project it onto many more species and individuals. And so this is where the frontier is now, is making those connections between those. And we have a couple software packages that are starting to deliver that and are essentially that kind of next generation of G2P tool that's available with GeneCAD and PlantCAD. And what's been most satisfying is we over 20-some years ago, helped use association mapping to identify one of the causal variants behind sweet corn. We can now go through with these large language models and recapitulate that in a few hours. So essentially, the tools and the power of those tools are truly allowing us to have amazing insights. And we're also now looking at able to take these tools and look across so many different species. Now we're looking at over 700 different grass species and their genomes in order to make the connections there. And again, we're relying on the whole series of tools, whether it's genomes in those environments, those machine learning tools to characterize the genetic variation, and then phylogenetic mixed models, and then it's allowing us to look at conversion adaptation. So those are essentially the tools we now are working with. And I would highlight what we've been able to do across the entire community is essentially create tools that are allowing us to, that have not only been powerful in maize, but they're also incredibly useful and powerful in all sorts of other species, whether that's mapping in humans to being applied to virtually every other crop and animal system in the world. So I now want to focus on what we've been able to use these tools for in order to really understand the story of maize and its genomics. And essentially, of course, like any species, maize is the product of its evolution in history. So let's talk about what happened during its evolution in the grasslands, and then how it moved during domestication, and then adaptation to North and South America, and then its evolution to modern production. So during those grasslands periods, it's essentially going to inherit this amazing photosynthesis, an adaptive genome, and a very strange seed. And the first thing I want to remind people about is when you went to middle school, you probably were taught the parts of a flower, and it probably looked like something like this. Flowering plants are animal pollinated with showy flowers and fruits. What they probably didn't emphasize was that grasses don't look like that at all. They're wind pollinated with reduced flowers and fruits and look very different. And so I think even though we're, as maize geneticists, very used to thinking about grasses, they really are strange angiosperms. Yet that strange angiosperm has been able to dominate about 40% of the planet today. So they're the dominant species on about 40% of the Earth's surface. But that didn't happen quickly. Grasses started evolving over about 65 million years ago, but grasslands themselves don't become common until the last 20 million years. And that mostly happens as CO2 levels are starting to drop, temperatures are dropping, and things like fire, ungulates are evolving, and C4 evolves. And then you start having these dominant grass ecosystems across the planet. And that's the inheritance that maize is having. And key to that was the C4 photosynthesis. There's a number of different varieties of it, but essentially the type that maize has, NADP-ME C4 photosynthesis, is an incredible evolutionary invention. And maize is able to essentially become the master of producing starch, but its related species like Miscanthus is a master of biomass, sugarcane of sugar, and sorghum is able to do those same types of things under even drought conditions. While not only they're great at photosynthesis, they're amazing in terms of their nitrogen efficiency and water efficiency. So essentially, maize is inheriting this great photosynthetic system. And so that's what inspired this group in the Penn Andropogonae project to start looking at all the Andropogonae. And so just the Andropogonae are the dominant species on about 17% of the Earth. And they've evolved these really diverse environmental adaptations. And so as you look across the Andropogonae, one of their keys to success was polyploidy. In the species we've sequenced deeply, we cataloged about 14 different events of polyploidy. And what you see here is that Zea's pattern, which I think we're quite familiar with, talked about a lot, it was one in which there was chromosomal fusion, substantial gene loss, and TE amplifications. When you look across the rest of the Andropogonae, there's much more stable chromosomes, minimal gene loss, and the TEs are unaffected by it. And so there's something going on different that's epitomized by maize versus the rest of the Andropogonae. And what I think we're seeing is that polyploidy is really about both managing load and expanding adaptive potentials. And while I put them as exclusive, I think both these things are happening here. Maize and Zea are showing that enhanced adaptive potential, while we need to add heterosis back in to manage genetic load. Other species are managing the genetic load through their polyploidy and expand their space. So when we start looking across all these species, what are we seeing? Well, it's thousands of genes that are involved at the coding level. When we look at genes associated with temperature and precipitation, hundreds of genes there, divergent nitrogen adaptations, and then again, hundreds of genes being involved in the shift from perennial to annuals across these. We also see the regulatory changes. About half the regulatory space in each of these ones are adapting as they adapt to each of these different environments. And we are consistently seeing rapid regulatory turnover. One of the things that's most striking is also the nature of a grass seed. And what I want to highlight here is that grass seeds have evolved to be poor nutrition for animals, because essentially they became a dominant clade, and they were facing off against insects and things like these ungulates like bison. Grasses started out with something that had more of a fruit like this you see on the left. And over that 65 million years, they evolved seeds that, in the case of maize, are 60% prolamines, which are very strange proteins. They're alcohol soluble and have very low levels of lysine. I think the key thing to remember about lysine, it's an essential amino acid, even for insects, and it's key for trypsin digestion. And so if you look at a maize seed or the grass seed, they have these prolamines, very strange proteins, which are totally different in their lysine content from every other protein in a plant. And those seeds are by essentially the product of a long-term evolutionary pressure. So maize has inherited amazing photosynthesis, an adaptive genome, and a strange seed. So when we talk about during domestication, what goes on is really maize is concentrating starch. And I think the one thing for us to kind of remember here is that eating grasses is really strange for primates. There's 400 or 500 different species of primates, but essentially only 2 of them eat grasses. And we're one of them. Essentially, over 50% of our diets are effectively now grasses. And so that's a really strange thing to do, but it's very successful because grasses were becoming the dominant species across most of the world's ecosystems. And when we look at those domestication genes that people like George Beadle were first identifying, and then John Dobley helped clone, those were essentially really about creating a harvestable sink, taking that starch and getting it into something that humans could harvest in an easy way. But when we started getting the tools of genomics, we were able to start really cataloging how many genes it really took to create that large ear that we have today that really concentrates starch. And we were starting to see it was over 1,100 genes were involved in that initial domestication phase, and then maybe 700 more genes were involved in latter types of adaptation. We were able to take a look at those early starch pathways, and essentially everything involved in starch production in maize was under selection. And we were able to even use ancient DNA studies to start asking when did that selection start occurring. And there was major selection for both starch and decreasing protein levels going back more than 4,000 years. So this has been a long-term selection that has been going on. And of course, although early on people were selecting very much for more starch, in modern times, now we have people who don't have enough various types of nutrition. And so we've been able to contribute using some of that maize diversity to increasing provitamin A levels. And one way to kind of think about that is we're adding the fruit back to this starchy crop. And those have been delivered by groups like CIMMYT and others as varieties that can be bought in East and Southern Africa. And you can even go to Amazon and buy grits that have these. So domestication was really about concentrating maize starch. Let's see what we kind of learned using these amazing genomic tools in terms of adaptation. Well, adaptation is not only about adapting to all the different rainfall and temperature environments across the Americas, but it's also adapting to the new agronomic environment that maize now grows in this high density species. And I think one of the most amazing things that came out of the NAM project is we took the world's diversity of corn, we crossed it, and even though flowering times differed in terms of a month or more, essentially the largest effect alleles were still measured really pretty much in hours. 98% of all the alleles had less than a 24-hour effect. And it wasn't just for flowering time. Essentially, when we look at leaves, 30 to 40 different genes control each aspect of these, and their individual effects were small and modulated and could be identified in populations like this. And so how do you get a lot of adaptation? You add up a lot of different alleles. And every line is pretty much carrying both positive and negative alleles. And essentially, that sum of those determines the adaptation. As we've been able to start getting more and more powerful genotyping approaches, we've been able to use, for example, ARS's germplasm collections to essentially directly map and identify some of those genes. And when we were able to look at over 2,000 inbred lines, we were able to identify about 50 regions controlling maize flowering time. But when you started turning to land races and both using trait and environmental GWAS, and you had 5,000 lines, you're starting to see more than 1,000 regions contributing to flowering and other types of traits. So the polygenicity of these complex traits is really high. So we were also able, though, to ask the question, what regions of the genome matter? And it turned out that most of the genome, 90-some% of the genome probably doesn't matter that much, but about 3% of it does. And it's pretty much, its impact is pretty much equally split between coding and non-coding regions in terms of the amount of genetic variance they're explaining there. And I'm still also struck when we look back and we did mapping on things like plant height. The largest effects we saw in NAM, for example, were only about 4 centimeters in size. And it has these hundreds or thousands of genes likely controlling the trait. But just because it's complex and it's controlled by hundreds or thousands of genes doesn't mean you can't predict it. And that's what genomics has really, and genomic selection and genome-wide prediction has been able to show us. Just because something's complex doesn't mean you can't predict it. And here's a beautiful example of that. You can add up all those effects that you see in NAM or the ARS diverse breeding lines, and essentially you can still predict with high levels of accuracy. And this is at the heart of why approaches like genome-wide prediction and genomic selection work, is those small effects can work together. So I want to just stop here and remind people that standing genetic variation is very different from new mutations. It's lots of small additive effects for most adaptive traits. It can be highly accurate and prediction is possible. We see very modest two-locus epistasis and modest levels of pleiotropy for a lot of the standing genetic variation. And the second thing I want to highlight is that environmental GWAS, which I think a decade or two ago, we were unsure how well this would all work. But if you just ask the question where various varieties of maize are adapted and you map the genes that are controlling that, it works. You can just see from, if you have well-adapted varieties, map the genetics behind there. And we've been starting to try to turbocharge that by looking at a highland adaptation across about 3,300 land races and then combining it with whole genome assemblies. And here we're seeing over 1,300 different adaptive loci were resolved down to individual genes and whole genome sequence behind it. And when you dive into what's controlling each of these, what you're seeing is that CDS regions and coding regions are about 6-fold enriched. They're being supported likely by proximal regulatory changes behind them. But going back to that polyploidy question of adaptation, the retained duplicated genes were even more enriched. So it's a combination of getting, having a couple copies of a gene, getting the right protein, and then supporting that with differential expression. So I think those were a lot of the lessons we learned about how maize was adapting globally. And now the question is, what have we been able to see using some of these tools in modern production? So essentially, the way we've gotten this amazing yield curve that we frequently show is essentially, of course, at the beginning, it had to do with heterosis. That's only the beginning of it. It then was turbocharged by an addition of fertilizer into the system, precision agronomy. And each one of those, though, were supported by genetics for disease, adaptation, density, and reducing genetic load. And together, that's what gives you that 7-fold increase in yield. We know some of that density work. Don Dubik was the one who really highlighted its importance. But then with some of these various populations and a lot of further studies that went on, we've made progress in understanding how density kind of works at the leaf level. But there's still, I think, much more to be learned as we look underground. When it comes to heterosis, I think we are starting to get a better idea, or actually a fairly good idea, of where it's coming from. And it's really an emergent property of two common response curves. The one you might want to call is the dominance one, where there's you get to a higher enough gene activity, and above that, you're in the robust range. And then there's a dosage curve, where you need to be between 2 points in terms of your gene activity and you're in a high dosage place. But essentially, heterosis is all about getting the vast majority of your genes into these two response curves. And we've been able to start seeing some of that effect as we did inbreeding and various types of other types of mapping. Essentially, what's disrupting this is about 5 new mutations per generation are disrupting this. We can see that interbreeding efficiency is reflected in recombination, where essentially we're having trouble purging some of those deleterious mutations. But essentially, as we got better and better genomics, deleterious mutations are everywhere. And yes, they do build up near low recombination regions, but they're everywhere spread across the genome. They're not only in encoding regions, they're also in regulatory regions. We've been able to essentially use profiling of mRNA levels and look at deviations there to show that about 20 to 30% of important traits like seed weight could be explained by these types of dysregulation that is occurring in the genes. But the really exciting thing is that we can now use tools, both evolutionary constraint to ask the question, does this given gene matter? We can combine that with these machine learning large language models to ask the question, do individual residues matter? We combine those together with various machine learning models, and we can start identifying the 100 to 200 deleterious mutations that may be in a given line. If you combine those two together, you can start improving genome-wide prediction for complex traits like yield by about 25%. And so I think the main drivers to success in modern breeding today is genomic selection, but it can then be supported by these other tools such as CRISPR, editing transgenes, our modeling of the central dogma, and crop growth models. I think all can play in very important roles to making this cycle move as fast as it possibly can for not only maize, but other species. So that's what got us to a high-yielding maize of today. I think the question is now, how do we design a corn for the 21st century? And a lot of these ideas are what we are combined together in what we call the Circuit Project. And so I showed you this plot before, and the key one I wanted to highlight here is that it was non-genetic were these nitrogen inputs. And they have been key to essentially the global shifts around the world. But we've been manipulating nitrogen for almost 7,000 years. It started out with crop rotation and manure systems. Then a little over 100 years ago, synthetic fertilizer becomes more common. And it drives the growth in population. Yet it also provides the vast majority of people today have food security. And that's because of nitrogen. That essentially nitrogen added to the food system helped reduce childhood mortality from an average pretty much around the world, nearly 50%, down to where we are today, about 4%. But it had unintended consequences, such as water pollution and greenhouse gas emissions. And what nitrogen 2 is about is really focusing that synthetic nitrogen and combining it with circularity to essentially address those issues. So in our current system, we fix nitrogen using Haber-Bosch, but only about 12% of it ends up on the consumer's plate. And essentially, the rest of it is being lost throughout the rest of the system. From a farmer's perspective, that means that the $10 to $20 billion they invested isn't getting to the consumer at the very end. It's 75% of U.S. water pollution and about 10% of U.S. greenhouse gas emissions. And so 2/3 of the nitrogen is moving along this path. And the question is, can we shift this? Are there ways we could re-engineer it? And the first thing that we can do is take that synthetic nitrogen and give it to the processes in the animals that actually really need it. And there's all sorts of ways of either providing urea directly, microbial biomass, enhanced silage, or amino acids. All are already used as supplements, but if we were to increase them from 3 to 5-fold from their very low level today to those levels, that would set up a massive change on what we can do on our cropping land. Essentially, we could reduce the amount of protein we pull off that land and recycle the nitrogen on our cropping lands. Combine that with more efficient recycling of nutrients through manure processing, and you end up with a system that requires about 50% less fertilizer going onto our industrial cropping systems. And that would also end up produce a system that is anywhere from 30 to 50% efficient over the one that was only 12% efficient. So, and what our predictions are, and the modeling is suggesting, is this would reduce fertilizer input costs by, would make it more than 50%, reduce those by 50%, water pollution and greenhouse gas emissions, again, by 50% or more. And I want to highlight that maize really epitomizes the food system's both logic and anomalies. Remember, it's this amazing grass at fixing carbon, and plants themselves, because CO2 is at a relatively low concentration in the atmosphere, leaves are wonderful ways of grabbing that CO2. While industrial chemistry, because nitrogen is quite common, but it has this very strong bond energy, is superb at breaking the nitrogen bond. Yet, these grasses our diets are made up mostly of grasses, yet they have this poor quality protein. And in the case of maize, we have this tropical annual that we're growing on a temperate grassland. And so the Circa project is really about addressing those problems. And at the heart of it, because corn is over 50% of the US nitrogen cycle. And so this great team has come together across the country to start working on it. And there's actually dozens more groups that are starting to collaborate with each of these great scientists and their great teams across the country. And so remember, corn has this great photosynthesis, uses that fertilizer, yet it has these two key anomalies. The seed is really not matched to modern uses, and it's this tropical species growing in a temperate environment. And so when we start walking through what we saw in grassland evolution, we ended up with all those prolamines in the seed. Then through domestication, we were trying to get more and more starch. That resulted in a seed that's 10 times larger. And even though protein concentration dropped by 50%, that still ended up that maize has 5 times more protein per seed than the beginning of domestication. And then through modern production, we now have this over 1 trillion kilograms of grain being produced, but over 100 billion kilograms of low quality protein that's being produced there. That's a lot of nitrogen coming out of the system. The second issue is essentially we plant corn in May, but essentially it's not covering the field till June. And essentially we have this reduced light harvesting window in these temperate environments. And you can see that very visually here, where the U.S. Midwest has the world's most productive farmers, and they're not able to effectively use maize photosynthesis. But by mid-July, that same region is then the most photosynthetically active region on the entire planet. And the second issue that happens with that out of sync is that it's out of sync with the nitrogen cycle. So corn's taking up nitrogen along this curve here. But essentially, the soil starts releasing nitrogen beginning in early April. And so we have this essentially asynchrony there. And so the question, and that results in essentially nitrate leaching and nitrous oxide releases. And so what Circa is trying to do is solve these anomalies by enhancing cold tolerance so we get better synchrony, and then figuring out better ways to keep the nitrogen on the land and recycle it year after year and season after season. The main targets are to essentially get enough cold tolerance to be able to move planting 1, 2, 3, 4 weeks earlier than we are right now. If you get enough cold tolerance, reduce the base temperature a little bit there, you would radically improve yields by over 20%. And then if you can decrease that grain nitrogen content from 8% down to 4%, it expands the time the nitrogen can stay in the leaves, which also enhances total yield and keeps the nitrogen on the farm. And the third thing we want to do is stabilize the nitrogen on farm with these processes called biological nitrification inhibition. And what they're doing is they're exudates and compounds that plants release that essentially prevent ammonia from being converted to nitrate, which is frequently lost on the fields. So Circa is focused on these 3 separate trait areas of using spring better, leaving nitrogen on the farm, and stabilizing nitrogen in the soil. And essentially, we've done various forms of modeling. Actually, I'm just showing you one of the 3 types of modeling we've done on this. And essentially, they're all coming up with a very consistent result. If you combine early spring growth, stay green, and low nitrogen grain, we're predicting nearly a 30% increase in yield, essentially a nitrogen yield efficiency increase of over 50%, and decrease farmer input costs by 23%, and reduce all sorts of environmental causes coming off of cornfields. You can combine this with soybean, and essentially soybean, which is, of course, a great protein crop, essentially the complete system wins on all measures, except for a modest decrease in total protein coming off the field, which can be made up with those other approaches. So the exciting thing is, those of us working with maize diversity and have been studying maize for all these decades, essentially maize and its wild relatives have all the genetics that we're kind of looking for. And so the question is, how do we discover, how do we deploy it into these future systems? And so I think 21st century maize genetics is combining these amazing tools with our understanding of where we've come from to then try to design a new type of corn that's efficient, adapted, and high yielding. And so why do we do maize genetics? I think because we get to do really interesting, exciting science, but we can also have a global impact. And I just want to end there. And I've just been really blessed with amazing teams and collaborators. And here's a pictures of a few of my lab over the year. I'll be happy to take some questions.
Great, thanks, Ed. All right, we're ready to take questions. What we really want folks to do is to raise their hands, and then we'll call on you and you can unmute and ask your question. And yes, I'll start. I don't see any hands up yet. So Ed, my opening question is, how do you bridge the current gap between where commercial maize is now and sort of the idea of the circa idiotype? In other words, you can model this and so show theoretically, like if you change the base temperature, you'll get these effects, but you'll have to do some genetics to get there. And then the question always is, in the public sector, maybe our base genetics aren't good enough to really convince people that this is where we go. Kind of, how is your vision to try to do this in the best available, like XPVPs, or collaborating with industry, or how do you get there?
Yeah, I think it is really important that we do collaborate with industry, evaluate what modern hybrids are actually doing, not looking at their genetics, and so we're not violating anything there, but we need to physiologically understand what modern hybrids are doing. And then, in each of these different areas, you can incrementally walk to the space we're going. There's not going to be, just like we haven't seen any super gene for any major adaptation that we really care about. It will be a series of genes, a series of genetics and understanding that we can kind of point to. And I think in a number of these areas, we can create models of what the trait would kind of look like, and industry can essentially replicate that idiotype probably very quickly within their own germplasm and do it. I think there's a lot of these areas that people are concerned about various trade-offs. How do you avoid this trade-off versus that trade-off? Because everything in genetics and plant physiology, there's always some trade-off. And I think we just need to kind of show that there's various mechanisms that this route doesn't result in a trade-off that we find unforgivable and things like that. So, I mean, essentially, we can get this new nitrogen efficiency, but we have to give up something. The something that we have to give up is grain protein. There is no free lunch out there, and and I think some of that early planting, it's just as important as the equipment to plant earlier. And the agronomy there. So I think that, like all areas we've ever seen in maize genetics, if we think about the system, the whole system, I think we can move in lockstep over the coming decades.
Thanks. Before I get to the next question, Ruben's reminding me to tell people that this recording is going to be available both at Maize GDB website and at the Z Evolution YouTube page. John Hodge has the next question. Go ahead, John.
Thanks so much. Really enjoyed the macro evolutionary story. It was fantastic. It kind of has me circling around an idea that I've been wondering myself for some time now, where you did a great job of sort of like outlining the importance of genome evolution and what you sort of gain either through sort of incremental mutations or through whole genome duplications or gene duplications. But in a way, like, it's sort of like in art, like negative space can sort of mean just as much. I kind of wonder, like, have you looked in the sort of grand scale of like, angiospermae and tried to ask, what if grasses or what has maize just decided that it doesn't need anymore? Like, what paralogs go away? And in a way, does that sort of help canalize it where it can only go this route now because it doesn't have that dimensionality anymore?
Yeah, John, I think that's a fascinating question, and we've looked at the chloroplasts across all angiosperms yet. We have not done the analysis you're talking about, but I absolutely think that is the type of question that what has been, maize is a very weird angiosperm, or grasses are. And, I remind the group, you should sometimes think of them much more like a pine tree, right? I mean, they have gone that type of direction. And I think, what they've lost, they've gained a lot. They've also lost things. And there's not, we can't, for example, do what goes on in a soybean seed, the same thing that what goes on in soybean is not what's going to go on in corn and its endosperm and everything. So they each have their own strengths. And that's why I think, trying to make every species do everything, I think is the wrong thing to do. If you have C4 photosynthesis, let that plant make starch and let it make oil. And don't try to push it to be some super protein species.
Great. Virginia Walbott is next. Go ahead, Virginia.
Hi, thanks, Ed. Great talk. I wanted to get your reaction to the National Chinese Corn Improvement Program. It's 1,000 scientists, and their goal is to raise corn protein content from 10% to 12% in 5 years.
I think I've outlined what my perspective is on this, and I think that's as all I'll say right now.
Okay, thank you.
David Horvath, go ahead.
Excellent. So along the lines of the first question, so you guys are trying to improve the freezing or the cold tolerance of corn. But corn was, originally a tropical plant. Do you think there's really enough genetic variability in the population to make a solid shot at really getting cold tolerant corn?
Yeah, David, I think that's a great question. And so, so I think that one of the things we've been able to see is that Tripsicum, is already adapted to pretty much growing all the way up to Wisconsin and things like that. So we have a sister genus, fairly closely related. It does a lot of the things we kind of wanted to do. We also see Miscanthus having photosynthesis in the time period that we wanted to do. That is where we needed to be. So I think the fundamental parts are there. The real issue is that Andropogones evolved in the tropics. And, I think if we were asking this question another 10 million years from now, this may have already been solved. Okay, it's kind of that we're asking this question right now when essentially the Andropogone and maize's lineage has only been in this space for 1 million or so years, and we need to now help it out. Okay, steal some ideas from other species, figure it out, and and I'll highlight that even shifts. We're not modeling saying we're going to try to get down to wheat level performance. Okay, if you can split the difference between where maize is right now and wheat is, and essentially you put the base temperature at half that, that's where you get these massive gains. So we're not trying to get to wheat, but I think we can do better than where we are. And those are the types of things that will be incremental, but those increments will be a couple percent, could be a couple percent of yield at a time. So they'll be meaningful.
Okay, Shujun, you're up next. Go ahead.
All right. Well, thank you, Ed, for the great presentation. My lab at Ohio State learned a lot. So I'm going to ask a question that hitting the Mexican top price topic, transposable elements. So since maize genome is 85% repetitive, with 80% of the genomes are transposons, I asked a question, same question to you earlier years, and now I want to ask it again. Like, how do you see transposons fit in the story of yield, seed development, starch, protein, maybe cold tolerance, nitrogen efficiency? How do you see it fitting into these kind of functional aspects of the maize genetics? And especially with the development of long-read sequencing, we can see transposon variation much better than short-reads now. Have you ever tried to look at it? And I want to get your perspective again. Thank you.
Yeah, well, thank you. Yeah, I think the reason that maize has such dynamic transposons, dynamic genome, is because most of those transposons are very well behaved. Okay, they do not impact phenotype. Okay, that doesn't mean obviously as maize geneticists we used actually the poorly behaved TEs to go study gene function and everything, but that doesn't mean that I think the tools we're starting to get with both genomics and machine learning models and AI, I think are going to allow us to start picking out that small percentage that really does have a lot of impact, okay? And I think we'll be able to do that not only within maize, but we'll be able to pick out the TEs that are affecting gene expression, that are affecting the chromatin structure, and we'll be able to predict those. And I think that's really where the push is how do we search through those TE needles in the haystack issue, not all the ones that are pretty much quiescent and not having impact, because there absolutely are some that matter. And as even if only 1% of TEs matter, 1% of pretty much the whole genome is still a lot, right?
Okay, thanks. We have another question in the chat from Karen Bruce. Karen, I don't know if you want to ask it. If not, I will.
Oh, hey, this is actually Wes Bruce. I'm using my wife's account. How do you do? Yeah, fantastic talk. Very much appreciated. And I think I wanted to raise a point that we have some folks that are working on nitrogen fixing capabilities and trying to figure out ways to transfer this, and maize, I think, would be one of the targets. Would this confound or conflict with Circa, or would it support it, or could you find ways to make it work if they were successful?
Right. So I think in those areas where we have industrial agriculture, I think the land use hit of getting nitrogen fixing corn is not worth it. Okay, it's gonna have a substantial yield cost. I mean, there's some of the reason why corn yields 3 times what soybeans does is because of the cost of fixing nitrogen, and even perhaps more important than just fixing it, it's defending it and creating an environment that does it. And so I think it's in general across the world, it's not worth the land use cost to have nitrogen fixing. But if you still want to, I think that low protein aspect actually goes beautifully with nitrogen fixation, because then you're in a lower input environment, because now you're reducing the total nitrogen budget that needs to be provided. And so by keeping, if you can make only a minimal amount of nitrogen, and you can keep that in the leaves the entire time during grain fill, you will be able to produce more starch and oil than you would otherwise. And I think that's one of the key things is the grain is stealing that nitrogen from the leaf. And if you're in a nitrogen limited environment, you've just killed yield because of that. And that I think is the key issue is and I think this is where I'd like talk to a lot of my colleagues and places where have don't have a lot of fertilizer available to them. For example, in sub-Saharan Africa, I still think we need to do a lot of system modeling where it may be better to have a low grain protein corn paired, of course, with legumes. Okay. I'm not saying corn's, and this is where I think we don't want to make corn everything. Okay, corn's good at some things, and other species are better at other, like protein and things like that. And so I think those intercropping of corn and legumes is a great system, and let each one be what it's really good at.
Excellent response. Thank you.
I have one more question that still there's a chance. So if anybody else wants to raise their hand, but while I have Ed, this is, I think I asked him this. This is another way, like Shujin, like I probably ask Ed this every 5-10 years just to see where we are now. But I think you showed a slide. Now that you've looked at all these Andropogone species, that maize is sort of exceptional in its, I guess, its structural genome variation. So I'm guessing also in general molecular variation. I just always come back to this question. Why is it so variable? Like, how does it maintain that level?
Some cold stuff.
Variation.
Yeah, well, I think, in terms of, the studies we're doing across the Andropogone, in terms of straight molecular diversity, these grasses, if they're outcrossing, they can maintain large effective population sizes. So from a diversity point of view, remember a lot of these grasses, I mean, every year we grow 10 trillion corn plants on the planet. And a lot of these grasses, when they were wild, also had high levels of diversity. So I don't think maize's diversity is super surprising for a clade that dominates 40% of the planet. Okay, it's kind of sitting where it should there. And, but that also means because there is so much diversity, and I think in the case of maize, it, in certain regards, we sometimes comment to one another. It's really kind of strange that maize is the species that took off and became the one that around the world, where Trypsicum dactyloides actually had a lot more environmental range of adaptations. But I think that, of course, had to do with annuals or respond to selection much more easily during domestication and so on. And so that was part of it. But that pre-adaptation, I think, of maize coming out of Mexico with tremendous altitudinal variation was able to preserve a lot of functional diversity through all the glaciations during the Pleistocene. And I think that pre-adaptation, it helps explain why it was super successful. So total high diversity, I think, is pretty common. I think combining that with the altitudinal variation you see in Mexico is probably the second key thing for maintaining things during glacial cycles.
Okay, we have a couple of our European colleagues are posting questions. Roberto, I don't know if you want to ask directly.
Oh, sure. Can you hear me?
Go ahead.
It's a great honor, Ed, and great talk as usual. Perhaps a silly question. Do you think we should invest as a community a little bit more in the roots down below in the soil, particularly in terms of exudates, or do you think mission impossible? What do you think, Ed?
No, I think we absolutely should work on roots too. I do want to highlight that I think plant breeders, when they select for density and group selection, I mean, I think that we do need to remember that when we select, when breeders select on a 4-row plot, okay, and they select for yield, they are selecting not only on the things we can see, but they are also selecting for roots. Okay,
absolutely.
So I want to make sure for everybody kind of realize just because breeding got started being done the right way in maize, and so, but I think we would learn a tremendous amount about what selection actually did happen during those periods. And I think there's still a lot to be gained. And I can, we can kind of paint that picture of what happened during domestication above ground. I'm not at all as sure what happened underground. I'm sure it's well adapted, but I'd love to know, and maybe that would provide some more of these intuitions on that. And then on the exudate side, and I think this is where we, again, everything has a cost, okay? And yes, you can exude lots of things. Maybe we were hoping the B&I chemicals, maybe they don't need to be super costly, but aluminum tolerance, for example, that's got a real cost exited, okay? And you lose things like yield with it and things. So I think we want to learn what's going on there, but I wouldn't want to, without good genetics modeling, say whether or not we'd want to do that.
Thanks, Ed. Great talk.
Great. We have Phoebe, I see you. You'll be the next question, but first I want to go to Alain Charcosset. He had a question in the chat. I don't know if you want to unmute Alain and ask.
Yes, thank you, Ed. Congratulations first. I guess I know the answer, but still I'm curious about your views on that, about QPM maize. If we would switch massively to QPM, would that to some extent change the reasoning that we should go for low protein maize?
I think QPM in places where people are directly eating the grain make a lot of sense, okay, or a lot more sense. I still think we should do the modeling, the tests, and the experiments as to whether or not pulling that much nitrogen out of the ground is a good thing from overall yield. I mean, for example, in lots of sub-Saharan Africa, the soils are such that essentially there's a lot of nitrogen mining going on. There'd be more productivity to recycle a lot of that nitrogen, and I think that's where the real whole system level question really matters. In the case of the US, we have lots of ways we can get nitrogen to our livestock outside of QPM that are super efficient. Anytime anyone wants to talk to me about how you can actually grow a cow with 100% synthetic nitrogen, I'm happy to talk about it. I've learned all about that in the last couple of years. But I think QPM is, in certain use cases makes sense, but I don't think it should be a broad spectrum thing that we want to use, because again, we're just not leaning into what corn is really good at.
Okay, thank you very much.
Phoebe Baker, you're next.
Hi, Ed. Thanks for the great talk. It's really great to have this opportunity to speak with you. I'm just wondering, as you brought up the aluminum toxicity, to what degree does improving corn's nitrogen use efficiency have to do with its interactions with other nutrients and maybe salts? Do you study that?
We have been looking a lot at phosphorus, okay, and phosphorus in the grain, but we have not been looking at some of the other nutrients, and we certainly in our circuit project have not been looking at the interaction between aluminum tolerance and this nitrogen efficiency, because we're targeting the U.S. Midwest, where that's not a big issue there. I will highlight the aspects that we see about phosphorus, and essentially the evolutionary reasoning that I was kind of arguing for. Maize has crappy grain protein. The reason why the phosphorus is bound up in phytate is pretty much for the same dang reason. This is a way that a seed would protect its phosphorus so it could be as useless as possible to any herbivore. Okay, so evolution has really worked hard to take those two most valuable nutrients, N and P, and make it as inaccessible as it possibly can be so that herbivores don't consume it. And so I think some of the same logic that we're talking about here on nitrogen does apply also to phosphorus. Having a maize grain with low levels of phosphorus makes a lot of sense, and then integrating phosphorus, if you're going to livestock through other routes, is likely the same type of process. And then you can recycle the phosphorus on farm. Which I think that's pull off the thing that is you really want off the field. Don't go through the plant if you don't need to go through the plant.
Okay, thank you.
Okay, we have a question from Pedro Revilla.
Hi, Ed. Thank you for this great talk. What do you think about these modern techniques, particularly about genomic edition?
I'm sorry, I didn't. The question was.
What do you think about using genomic edition?
Genome editing type of things. Yes,
No, sorry.
and yeah. So I mean, I think it's an amazing technology. I think it has a lot of potential. But and actually, there's a nice review that Guillaume Ramstein just put out in Nature Reviews Genetics that I think covers kind of our perspective on it. And I think we just need to ask the question, where does it fit into a GS system? Because essentially those edits have to be able to compete again in terms of time and cost against genomic selection. And I think the way that paper breaks it down is that we can use this knowledge of causal variance both in precision selection or in editing. And so in either situation, you've got to make a decision as to what's going to be most cost effective and efficient to move things forward. And in some cases, if the allele is not in your close germplasm pool, it may absolutely be the right thing to do is to edit it in. And that's the fastest way to get there. And so I think it really just needs to be, where we use editing, where we use precision selection is very much, an economics effort analysis that needs to be done in each one. But it's great to have it as a tool. If we can start doing editing on 1,000 loci at once, those economics will change real quick. Okay, I think that's the issue: is tweaking one or two. Precision selection can compete pretty well. You start editing 1,000 things precisely, we're in a different, the economics all start changing.
Thank you.
We have a question from Jinliang Yang. There, Jinliang.
Can you hear me? Hey, Ed, congrats. So my question is about AI. So you're a pioneer of AI in maize genetic research. But my question is about your vision for the role of general AI, the software type of AI. Students are now using it for doing data analysis and even polish or maybe writing their manuscripts. I think that has changed our research community a lot. So what's your vision of this type of technology gonna impact our data research and impact on our community in general?
Yeah, well, I would, in this area, I would say my first answer is I don't know. Okay, is the honest answer there. I think we absolutely need to build tools and make our data in ways that's accessible, and so that AI agents can use it effectively. But I also think there's a lot of issues that the principles in which science work in terms of positive and negative controls, testing hypotheses, and things like that. I think we need to make sure we build into these AI systems. I think the ways by which we're going to have amazing ways to come up with new hypotheses and things, but I think that building rigor into how we use AI to test hypotheses, we really need to make sure those systems are there. And we do a lot of software development, and when you do software development, you're talking about unit tests. Does it pass this step, this step, this step? And in general science, those are positive and negative controls and things like that. But we really need to have established unit testing, integration testing of our science. And so we know we're standing on firm grounds on these things, and it's not a bunch of junk. Okay, that sounds right, but it's just, lots of things can sound reasonable but not be right, and it's sometimes those most non-intuitive things that you realize, oh yeah, that's where I have to lean into, because wasn't obvious.
Yeah, great answer. I'll say my experience with Ed has always been like, Ed is sort of the opposite of AI in that sense, where Ed will say things and then my immediate reaction is, there's no way that's true, right? And then 10 years later, obviously it was true. So AI does the opposite. It just sort of tells you the generally accepted thing and doesn't give you the room for creativity. So thanks, Ed, that you're not a machine. And there's lots of congratulations and people thanking you for all your work for the community. You've been a big impact on the community. So we'll try to download the chat session and you can see all the good wishes that people have sent to you. So
thank you.
Sorry for joining. Thank you so much, Ed.
Thank you all.
Thanks, Ed. That was great.
Yeah. So how much the first few slides or first time we were kind of all, what was going on?
It's I noticed when you tried at the beginning, it was it had like a very faint gray haze.
Oh, okay.
Yeah,
that was weird. You know,
we saw it. I was reluctant to ask you to stop, right?
Yeah, we could see it on our screen. That's the part that was confusing.
No, we could see you. It was fine. They had a like a.
It's good. It's good. My lab yells at me, so you know that's what always keeps me honest. Anyhow, right? Is my lab.
I'll try to on the recording. I'll try to splice that half minute out, so you know it looks good.
Okay, sounds good. Thank you guys so much for providing this opportunity and everything. Okay.
And Ed, you had there were like 400 participants, and many many of us are here sitting on rooms where there are many people. So
20 people right in this room.
Oh yeah, I wouldn't be surprised if you had something like 500, 600 people sent to you today. So.
Wow. Okay. Well, thank you guys very much for that opportunity. Bye, Roberto.
Ciao, Roberto.
Yeah, ciao. We need to buy you beer. We need a standing ovation for you, Ed, but we'll have an opportunity. If you happen to go through Bologna, come and visit, okay? It was great to see you.
Good to see you, Roberto.